Most parasites drain, weaken, or kill their hosts, but Sacculina does something far stranger. This barnacle invades a living crab, hijacks its hormones, and rewrites its instincts, forcing it to protect and nurture the parasite's offspring, even if it means losing its own future. What twisted biology turns a crab into the perfect babysitter for its enemy? The answer starts deep inside a seemingly healthy shell. Sacculina looks nothing like the barnacles clinging to ship hulls or rocks in the tide. Yet, at its core, it is a barnacle, a crustacean, just like the crabs it invades. Most people expect parasites to be worms or tiny insects, but Sacculina belongs to a group called Rhizocephala, a lineage of barnacles that abandoned the hard shell and filter-feeding lifestyle of their relatives. Instead of growing a protective armor, Sacculina has evolved to become almost invisible, both to the eye and to its host's immune system. The journey begins as a free-swimming larva, drifting in the ocean currents among plankton. At this stage, Sacculina shares features with other crustaceans: jointed limbs, a segmented body, and a tough exoskeleton. But this familiar form is temporary. Once the larva finds a crab, it undergoes a radical transformation. The outer shell is discarded, and the parasite sheds nearly every trait that marks it as a barnacle. What remains is a tiny, specialized cell mass equipped to infiltrate its host. This evolutionary path is rare. Parasites that attack their own close relatives are the exception, not the rule. Sacculina's entire existence is built on this betrayal of kinship. It is not an invader from another branch of life, but a crustacean that has turned on its own. The result is a parasite that defies expectations, not a worm, not a fungus, but a barnacle that has traded its shell for a hidden, predatory life inside another crustacean. Green crabs haunt the rocky tide pools and muddy estuaries of coastal waters, their bodies armoured in mottled green and brown. These crustaceans belong to the order Decapoda, a group defined by 10 legs, quick reflexes, and a hard exoskeleton that must be shed to grow. Each year, a green crab undergoes ecdysis, a molt ritual where the old shell splits and a new, larger one hardens in its place. This process is essential for survival, but it also leaves the crab exposed and vulnerable, a brief window when predators and other dangers can strike. In their normal state, green crabs are active and opportunistic. They scavenge for food, hunt small invertebrates, and sometimes even prey on smaller crabs. Their sharp claws and keen senses make them formidable hunters. Beneath the shell, the crab's reproductive system is tuned to the rhythms of the sea. Females carry fertilized eggs in a dense mass tucked under the abdomen, known as the sponge, while males compete for mates and defend territory. This cycle of growth, competition, and reproduction shapes every aspect of crab behavior. A healthy crab's life is a constant balancing act, finding food, avoiding danger, and preparing for the next molt or breeding season. The abdomen stays tightly folded, protecting delicate gills and reproductive organs. Each limb and muscle is built for speed and defense. The crab's world is one of instinct and routine, every movement honed by millions of years of adaptation. But this ordinary life is built on fragile foundations. Any disruption to the crab's ability to molt, feed, or reproduce can unravel the entire pattern, leaving the animal exposed to forces it cannot resist. The cyprid larva, now stripped of its armor and identity, clings to the crab's shell and begins its most invasive act. It morphs into a kentrogon, a syringe-like life stage, built for only one purpose. At a vulnerable joint in the crab's exoskeleton, the kentrogon deploys a needle-thin stylet. With surgical precision, it pierces the armor and injects a cluster of living cells into the host's body cavity. The shell and limbs of the parasite are discarded, left behind like a spent casing. What enters the crab is a shapeless mass, but within days it unfurls into a network of root-like filaments called the interna. These tendrils snake through the crab's tissues, wrapping around organs, weaving into the nervous system, and tapping directly into the crab's blood supply. The interna is not just a feeding structure, it is a command center. As the network spreads, it begins to sabotage the crab's internal chemistry. The parasite halts the production of gonad-inhibiting hormone, or GIH, the signal that regulates reproduction. At the same time, it hijacks the host's vitellogenin, a protein normally used to build egg yolk, redirecting it to support the parasite's own needs. The crab's reproductive system is dismantled from within, its energy reserves siphoned off to feed the growing parasite. Molting, once essential for growth and survival, is now impossible. The crab's body is frozen in its current form, unable to shed its shell or regenerate lost limbs. Every cell, every pathway is slowly reprogrammed. The parasite's root system does not simply drain the crab, it rewires the animal's biology, turning the host into a living vessel for its future offspring. A pale, swollen sac now hangs beneath the crab's abdomen, an alien structure called the extern. This is the parasite's brood chamber, packed with hundreds of Sacculina larvae, each one destined to continue the cycle. Where the crab's own eggs would have clustered, the parasite's offspring now grow in a fleshy pouch tethered directly to the host's body. The crab, stripped of its ability to reproduce, becomes a full-time caretaker for the parasite's brood. Every day, the infected crab tends to this sac with a devotion that borders on obsession. It grooms the surface, picking away debris and algae with careful strokes of its legs. The crab's abdomen, once tightly folded, now arches outward to cradle the externa, providing maximum protection. The parasite has even widened and flattened the crab's abdomen, reshaping the host's shell into a perfect nursery platform. The crab does not simply guard the brood sac, it aerates it. By rhythmically pumping its abdomen, the crab flushes fresh seawater over the externa, delivering oxygen to the developing larvae inside. This behavior mimics the way a healthy female crab would care for her own eggs, but here, the instinct has been hijacked. The parasite's root network has reprogrammed the crab's nervous system, ensuring that every action serves the needs of Sacculina's young. If danger approaches, the crab shifts its body to shield the brood sac, placing itself between the externa and any threat. The crab's own survival, its drive to feed, even its urge to molt, all have been sacrificed. What remains is a single, overriding purpose: to protect and nurture the parasite's next generation. Male crabs are not spared from Sacculina's control. Once infected, a male's identity is rewritten from the inside out. The parasite's root-like network suppresses the androgenic gland, the organ responsible for producing male hormones. Without these signals, the crab's body shifts course. The abdomen widens and flattens, taking on the shape of a female's. Even the crab's behavior transforms. He begins to cradle and clean the parasite's brood sac, mimicking the maternal care of a female crab. The external signs of maleness vanish. What remains is a body and mind repurposed for a single task: tending the parasite's young. This forced feminization is not a minor tweak. The crab's own reproductive organs are destroyed, and its instincts are replaced. The male carries out elaborate brooding rituals, pumping seawater over the externa and defending it from threats. In the wild, entire populations can be affected. In some regions, up to a quarter of green crabs show signs of Sacculina infection, their reproductive futures erased. Fisheries managers have debated using Sakhalina as a biocontrol agent against invasive green crabs, but the parasite's broad host range threatens native species as well. Sakhalina's strategy is brutally efficient. Rather than killing its host, it preserves the crab's body as a living nursery, extracting every possible resource. This approach is echoed in other parasites, like Toxoplasma, which manipulates rodents to lose their fear of cats, ensuring the parasite's transmission. Across the animal kingdom, parasites that rewire behavior outcompete those that simply destroy. Sakhalina stands as one of evolution's most extreme examples, a parasite that erases its host's identity, all for the sake of its own survival. Right now, biologists estimate that up to 30% of some wild crab populations are under Sakhalina's control. As climate change disrupts ocean ecosystems, the reach of such parasites may only spread. In nature, control is rarely obvious, and sometimes it wears a familiar shell. Thanks for watching.